Hello, and thank you for joining the Thomas Expert Series. We're going to get started in just a minute. In the meantime, I would like to ask everyone to please mute their microphone. This expert series will be recorded and on demand within 24 hours or so. So please let your colleagues know how much you enjoyed the presentation today and send them to the on demand tab. To get everyone a little familiar with the chat, where's everyone tuning in from today? I'm in New Jersey. Patricia, where are you at?
I'm New Jersey as well.
Oh, nice. Got another South Jersey person in here. Got Philly. Perfect. All right, so I'll go ahead and turn it over to Patricia here today to talk about Fighting for Food Safety, a guide to maximizing agriculture and food safety testing.
Thank you very much. So today we're going to talk about food safety, how you test for it, what you test for it, and how do you prepare your samples in a way that you know that you're going to get the best, most accurate results that you can. Oops, sorry, I jumped ahead of them. So if we're looking about worldwide illness and death, according to the World Health Organization, 1 in 10 people in the world will sicken or die of a foodborne illness. So that equates to 420,000 deaths a year. In the US, there are 48 million illnesses due to foodborne illness. That's 1 out of 6 people, and it amounts to about 3,000 deaths a year. So where do these foodborne illnesses come from? Well, they can come from natural or incurred toxins, things like cyanide in different seeds of different plants or trees or fruits. They can come from adulteration or counterfeiting, where somebody thinks that they're getting one particular type of material and they're getting something else, that it's been counterfeited or adulterated. It can come from process contamination, from cleanliness or how it's handled, or from the packaging. And these can be physical contaminants like bugs or insects or parasites, or it could be things like microbiological and chemical contaminants. And there are well over 250 different types or categories of foodborne and waterborne agents of disease. For microbiological targets, we're mostly looking at things like norovirus, and that's of course a virus, and it has about 125,000 cases worldwide a year. But if you're talking about a really deadly foodborne illness, look at Listeria monocytogenes, It's a bacteria, and it has about 14,000 cases a year, but it's got a death rate of about 22.5%. So even though there might not be a very high target for infections or worldwide cases, it doesn't actually mean that it isn't a serious threat to human health. So when you actually prepare your sample, your food sample for testing, you need to consider what's the temperature I'm preparing it at? Is temperature a consideration? What's the homogeneity? And is my sample homogeneous to begin with? Is it a beverage, or is it something that's very well mixed? What's its particle size? And how much time am I going to be preparing the sample for? And is that going to affect things like time and particle size and homogeneity? The first thing that you have to understand, that sample homogeneity means that it is on the entire batch or sample. When we talk about representative samples, that's slightly different. Representative samples reflect a population size, and it's the smallest subset of a population that still retains the property of the bigger population. So imagine that you're in a, let's say in an apple orchard, and they have lots of different apples and lots of different trees all over this apple orchard. You want to make sure that if you're sampling the entire orchard population, that you're representing all the different variations to some degree in your subset. And this subset has to be statistically valid, and it has to reasonably represent the population. Now, when you have that bushel of apples for testing, and you know that you have a reflective representative sampling, then you take that to the lab or to the processing area, and then you try to process a sample for homogeneity. This is where you process the batch or the sample, and you're trying to make it uniform in composition and character. With liquids, that's really easy. You shake, you mix, as long as they don't layer upon each other, you have high homogeneity. That bushel of apples, yeah, that's kind of low homogeneity. That's going to take a little bit more work. And the larger the particle, the less homogeneous the sample is. So, how small do you have to go? This is one of my favorite studies I've ever read, and it was about particle size and the amount of sample that you need for particle size. It was for talking about food for animals, the Feed Association. So the citation is there. Some of the take homes with it was that depending on what level of uncertainty you need in your calculations, in your results, you could drastically change the amount of sample that you need by reducing the particle size. So let's pretend that we get a sample of, let's say, popcorn. And popcorns are all about 5 millimeters for each popcorn kernel. And we say, well, that's good enough. I mean, we're just going to mix up this batch of popcorn kernels and, we'll be good for that. So that 5 millimeter popcorn kernels, we have this big bag of it. We're going to say, this is our sample. And we'll think this is going to be good for us to use as a sample. Well, if you are working in a 10% uncertainty, which is very generous, you need 125 grams of that sample. So you need a big old cup of that sample in order to say that you've processed it correctly. But if you take all those popcorn kernels and you grind them down to basically dust, or you grind them down to flour, now you need 0.13 grams of sample. If you have a very tight uncertainty at like 1%, you need a whole lot more. You're going to need 12,500 grams at that popcorn size, or at the flour size or the powder size, you're going to need 12.5 grams. Also, the smaller the particle size, the easier it becomes for you to fully extract or digest a particle. If you have a large particle size, then you might not get to the center of that particle for an extraction. Or for a digestion, it might take longer to digest, but a smaller particle, you have a better chance of getting to the center of that particle. So now let's look at some of the other considerations. These really depend on what is your target. Is it a chemical? Is it a microbiological? Is it molecular biological? And what method are you going to be testing that for? So let's look at particle size. We talked about particle size a moment ago. Well, what's the range of particle sizes? Well, if you're looking at a fine tip pen, a 0.5 millimeter fine tip pen, that's 500,000 nanometers. You go down to beach sand, 100,000 nanometers. Grain of salt, 60,000. Talcum powder, 10,000. Now you start getting into the range of more fine analytical grinding, things like dust or mold. Some of the mold spores are 5,000. And you can see where the different types of equipment can grind, laboratory equipment can grind down to much smaller levels of particle size. What you do have to worry about, though, is if you're concerned about blood cells or mold or bacterium or viruses, if you go too small in the particle size, then you will actually kill your living cells. So you want to be very careful about your particle size in your cells. And you're also going to take some time to really optimize a grinding method where you're spending the least amount of time to grind to the particle size, because the longer you grind a sample, the smaller the particle size, but then you also start to get clumping involved as well. So you can have some physical changes to the sample if you overgrind it as well. And then chemically, if we look at particle size and chemicals, there are 3 types of scenarios that can actually make the decision about what particle size you stop at when you grind your samples. Now, for something like cannabis, cannabinoids, as you grind the sample smaller, your cannabinoids increase. So you break open all those trichomes, you break open all of those cells that have the oils and the terpenes and the cannabinoids in them, you get increased yield. But something like coffee or tea with flavonoids, Well, the flavonoids kind of, when you're in an aqueous solution, so you have that cup of tea in hot water, yes, the smaller the particle size, the more of an extraction. But researchers found that if they tried to do a solvent extraction, what they had was a sweet spot for the particle size. If you did it too small, you didn't get the best results, and if you did left it too big. So there was a sweet spot for something like flavonoids. With your coffee, think about caffeine. As you grind the sample finer and finer, you get more caffeine extraction. But when you have the aldehydes and the terpenes, it depends. There is a sweet spot for those aldehydes and the terpenes in coffee. When you look about microbiological pathogens, things like your norovirus and your listeria and shigella and aspergillus, they have relative sizes. And the larger the bacteria, the microbiological organism, the better the chance that if you're reducing particle size, that you have to be very careful at your stop point, because you could actually lyse your cells or you could kill your cells. And you actually may cause some preferential treatment by type or by strain. So if there are multiple different strains of a particular bacteria or multiple different types of bacteria in a sample, and some are larger than others, by over grinding the sample, you lyse the cells, you kill them. And if you're going to be doing anything where you require live cells, you've never now preferentially treated the smaller cells. So they're the ones that are going to live, and the bigger ones are going to die. We did a little study where we wanted to see the DNA extraction, and just overall when we were using it for micro and molecular biology, we found by automating your sample preparation, instead of doing it by hand, a manual homogenization, by using something like our GenoGrinder, you are able to get more DNA extracted from the sample. So this is an example of the quality and the amount of DNA we were able to get from different types of samples. And we have mouse heart, chicken breast, and beef loin here. But there are some alerts or some things you really need to think about when you are decreasing your particle size. As you decrease the particle size, it becomes an inhalation worry, or a possible safety concern. It could be flammable. Has anybody heard of when a flour mill exploded out in the Midwest? It's because, believe it or not, flour can be very flammable when it's aerolized, and all it took was a spark or a self-ignition, and suddenly you had fire. If you are doing any processes where you're going to have to filter, or the particles are going to have to pass through an area, it could gum up any gears if you're using a mechanical type of grinding unit, or it might stop up any filtration that you might be doing with it. Chemically, you can cause chemical reactions with smaller particle sizes. You can get oxidation effects. You could get hydrolyzation effects, where the smaller particles have more surface area and they pull in more water. So then you have hydrolyzation. And if you have anything that depends on live cells or are very volatile, you could actually volatilize those. Talking about live cells, you could, of course, lyse the cell, but you could also expose them to oxygen. You can cause oxidation. Now let's look at the other, temperature and time, and they are very much interrelated. The more you grind a sample, the more it heats, and therefore the potential for heat damage. There is a Goldilocks zone, or a sweet zone, for most living organisms, most cells, most biological organisms. And those tend to be in that range of 0 to 40 degrees C. So this is like the Goldilocks zone. That's where life basically thrives and exists. So if you take living cells and you overheat them beyond that, you could kill off the cells or living tissues. Also the same, if you're looking for living tissues and living cells and you cool them too much, you can kill them off. Microbes, they tend to have a little wider range. But again, if you start to heat them up too much, you get some protein denaturation, and you start to get the breakdown of living microbes. DNA, again, you get the degradation of proteins as the sample heats, and they'll denature. DNA, though, does pretty well with cold. So cold and sub-cold, sub-freezing temperatures are usually fine for DNA. And chemicals as well. They have a tolerance. Most chemicals have a tolerance for cold. It's the heat, some of the volatiles and the semi-volatiles, that you start to lose. So when we look at temperature and chemistry, increases in temperature increase reactions. It increases dimers. You can make different structures. You can get different interactions. You can volatilize your chemicals, and you can cause chemical reactions. And if you take chemistry too cold, you can get ice crystals. You can get the accumulation of water. Whenever you work at very cold temperatures and you're exposed in room temperature air, you get the accumulation of moisture towards your sample. You can have different changes in structure. Sometimes freezing a sample will change its structure. And sometimes when you freeze a sample and it will unfreeze, you start to get layering or separations of samples. This all depends on how quickly you heat or cool a sample and how much time it's exposed to those temperatures. For pathogens, they have a maximum temperature and time, where if you overexpose that particular pathogen for more than that time period, you will kill that particular pathogen. So like Campylobacter, 70 degrees C, and if it's over 2 minutes of exposure, you'll kill that sample. Clostridium or botulism, over 85 C for more than 5 minutes, you will kill that particular microbe. So sometimes it really matters on, and it doesn't even matter sometimes if you are going to kill the entire sample or not. Let's say you're working with E. coli and your processes are getting pretty warm and you're getting at 60 or 65 or not even 65. Let's say you're getting about 50. You're not going to kill 100% of the sample, but you're going to kill some of the sample. We did a study of grinding under different temperature conditions. We used our little Geno light here, and this was just water. And what we found is uncontrolled. We put in a tube of water and we ground it for different seconds, 50 seconds, 100 seconds, 150. You can see that in 150 seconds, it already had hit 40 degrees C. And if we had continued longer than that, that temperature would have risen. So we employed then some cooling, some Peltier cooling on this, and we were able to keep the temperature steady. So there are going to be samples, whether they're chemical or microbiological, that you're going to need temperature control for. So you want to choose the correct sample preparation method for the type of materials you'll be testing. So something like cryogenic grinding, good for things like chemicals and DNA, not good for living cells and living microbes. Temperature controlled is pretty much good for anything, and something like our Geno grinder or our Geno light. Or an uncontrolled, well, that's going to be a time experiment then. So you're going to be dependent on how long you're actually processing your sample. We have a new product called the CryoBlade. This is sample grinding. It uses either dry ice or liquid nitrogen, and it grinds all different types of bulk samples: apples, strawberries, gummies, animal tissue. So this is very new to us. So if you want some more information, we'll be happy to provide it to you. Now let's see how this all fits together. Sample processing and optimizing your sample processing and preparation will help you increase your accuracy, speed, and efficiency. Then you optimize your method with your state, your type, your particle size, the time, and the temperature you're grinding at. And then you verify those with QCs and certified reference materials. Then you optimize and refine your method testing process with standards, including QC, standards, and PTs. Now, there are different types of standards you can use. You can use qualitative standards. These are pure materials or pure strains or pure chemicals without any known certified value. These are more like identity standards. You have quantitative standards. These are pure or blended strains, chemical solutions or materials or concentrations with enumerated values. These are values that you have on a certificate, like a certified reference material. Then you have a QC known. This is a reference material that's been retained from a PT study and assigned a value. For our microbiological CRMs, you actually have to know what you are trying to do with them. Are you going to do something like colony testing? Are you going to be counting CFUs? Are you going to be using most probable number or MPN? So you want to be able to match the microbiological CRM with the type of application that you're using it for. For instance, our pelleted CRMs for microbiology, they're good for things like Petri dishes and water quantity test applications or membrane filters or CFU calculations. And when you want to use one of these bacterial certified reference material, you first open the vacuum sealed vial and you reconstitute the pellet in an appropriate hydration fluid. And then you make your aliquots before you put them onto your agar medium. And sometimes you'll need to make multiple serial dilutions to get the appropriate bacteria concentration on your media plate. And then you'll incubate your samples, and then you will be able to record your results. We also have something called a snap stick. A snap stick has a pellet in the snap stick that contains live bacteria or fungus, and that it has to be rehydrated with the appropriate media. You rehydrate that, and then you swab or inoculate your streak culture, and then you incubate your plates, and then you are able to examine the morphology and the colony growth on your media. So we are a family of standards. We have our QCs, our PTs, and our CRMs. We have specs, which is the organic, inorganic chemicals. We have zeptometrics, which are microbiological and molecular biological CRMs. And then we have NSI, which is our chemistry, microbiology, and proficiency tests. So we believe that any needs you have for standards, CRMs, QCs, or PTs, we are able to provide it for you. And I would like to thank everyone for this opportunity, and I am ready if we have some questions.
While everyone is typing out questions and thinking about questions, I'm going to go ahead and share my screen for just a moment. So I just wanted to take a moment to show you guys that there is a survey in here that you are, or that there is a giveaway in here if you take the survey. And then in here, you will also find literature, web links, and videos. And be sure to hang around in the environment after the experts talk, and you can explore the on-demand talks, which are hanging out up here. And you can go ahead and explore the Thomas Center. And this talk will be on demand by tomorrow. So make sure you bring your customers and coworkers in here that you think will benefit from these talks. And I'll go ahead and hand it back over to Specs to finish us out with the Q&A.
Thank you very much. So I know I went a little fast there. Sorry, I'm losing my voice a little bit. I wanted to make sure I got it all in. So one of the questions we have here is, how much concern is there for plastics in food? Are there standards for plastic? That's actually a really good question. Plastics for food safety is a very big issue. And depending on the inks and the plastics that they use, there can be a whole sort of different types of contamination that could potentially cause illness from food. Whether it be inks, I think we all know about the infamous M&Ms and the red dye and things like that. But there's a lot of what are considered to be additives and packaging, anti-slip agents and things like that, that potentially could be a problem with packaging for food. So it's a very big field of plastic analysis when it comes to food. And there are standards, both chemical standards and physical standards for plastics, and potentially any chemicals or microbiological organisms or anything else that might be associated with plastic residues in food. Can I use the same process sample for both metal and organic testing? It depends on how you have processed the sample. If you use a sample processing equipment, a piece of sample processing equipment, you want to know what the composition of it is. If the composition of the grinding media is metal, if it's something like a bladed blender type of system, or you're using, excuse me, metal balls as grinding media, then if you are looking for inorganic targets such as chromium, lead, aluminum, and things like that, you might have contamination from the grinding media or from the blades of the sample preparation equipment. On the other hand, there are pieces of equipment. Our freezer mill has polycarbonate vials, and so that's totally plastic. So that's great for doing metals analysis. So if you're doing inorganic analysis, you can use polycarbonate vials for the freezer mill and you won't get any metal contamination. However, on the other hand, if you are doing something where you care about plastic potentially, then you might want to consider whether or not the polycarbonate vials are going to interfere with whatever your targets are. But in general, if you're grinding it for inorganics, for the most part, you can use it for organics, unless the plastic interferes. If you're grinding it just with metal components, then yes, you have to worry about inorganic contamination. I may have missed this, but do you have any cannabis testing products? We actually do have a full line of cannabis products, both on the sample preparation side. They've used the freezer mill for gummies and the Geno grinder and the Geno light for plant materials and other types of materials. On the CRM side and the PT side, we have a full line of proficiency tests for the cannabis industry, and we have a full catalog of cannabis standards as well. We also have microbiological standards for the cannabis industry as well. Anybody else have any more questions? Well,
I
thank
don't
you.
think I see anything. All right, everyone, thank you so much for joining Specs and Thomas today. I hope you all have a great rest of your day.